Hi. After transcription, the next important step in gene expression is the translation of mRNA into protein. Today we're going to discuss this topic in detail, and I've included some animations that show us what we know now about how the protein is actually made. So I hope you enjoy it. Please prepare questions for question time, and here comes the voiceover. Here's an image of the machinery that makes translation possible. This is the 70S ribosome from bacteria. It shows the locations of the mRNA and the tRNAs, and also the two subunits, the 30S, which is a smaller subunit, and the 50S subunit. Today we're going to talk about how the ribosome translate an mRNA into a protein. So in translation, proteins are assembled from amino acids during this process. The mRNA specifies as which amino acids are assembled into a polypeptide chain during translation. Translation occurs at the ribosome, which I just showed you in the last image. The polypeptide chain is the primary structure of a protein. Interactions between neighboring amino acids in the chain causes the protein to fold into complex 3D structures. The building blocks of proteins are the amino acids. And here we have an amino acid. And all amino acids share the same basic structure. You have an amino group, or carboxyl group, and then a radical group, the side chain. And this side chain is different for each type of amino acid, and it gives the amino acid special chemical properties. For example, there are amino acids that contain nonpolar aliphatic R groups, such as glycine, alanine, valine, leucine, isoleucine, and methionine. Others have polar uncharged R groups, such as serine, threonine, cysteine, proline, asparagine, and glutamine. Some amino acids have aromatic R groups, such as phenylalanine, tyrosine, and tryptophan. Others have positively charged R groups, such as lysine, arginine, and histidine. And of course, there are some amino acids that have negatively charged R groups, such as aspartate and glutamate. Since we are interested in translation, we want to know how we can connect amino acids to the genetic code, which has already been done. So I'm going to go through some of the experiments that got us here. So what amino acids are specified by codons composed of only one type of base? And the methods for this are pretty easy. You obtain a large amount of a particular nucleotide, for example, here, uracil. And then with a polynucleotide phosphorylase, you can make a poly-U homopolymer. The homopolymer is added to the test tube containing a cell-free translation system. One radioactively labeled amino acid and 19 unlabeled amino acids. The tube is incubated at 37 degrees Celsius. And under these conditions, translation can take place. The protein is precipitated and passed through a membrane. Free amino acids just pass through. Then the filter is checked for radioactivity. What you get when you do this is that one amino acid is radiolabeled. In this case, the procedure was repeated in 20 tubes, with each tube containing a different labeled amino acid. The tube in which the protein was radioactively labeled contained newly synthesized protein with the amino acids specified by the homopolymer. In this case, uracil, uracil, uracil specified the amino acid phenylalanine. So the conclusion is UUU encodes phenylalanine. In other experiments, AAA encoded lysine and CCC encoded proline. Before I can explain those experiments, I need to talk to you a little bit about tRNA, transfer RNA. Here you can see a cartoon of tRNA where you have the tRNA bound to a particular amino acid, and it also has an anticodon which interacts with the mRNA. So tRNA are recruited to the ribosome to provide amino acids during translation of mRNA. The specificity between an amino acid and its tRNA is determined by each individual amino acid tRNA synthetase. There are 20 different amino acid tRNA synthetases in a cell. tRNA charging is a chemical reaction in which an amino acid tRNA synthetase attaches an amino acid to its corresponding tRNA. So if we use tRNAs, what other matches between codon and amino acid could be determined? So here's what you do. You take very short mRNAs with known codons were synthesized. And here you have a synthetic mRNA with just one codon. And you add a mixture of ribosomes and tRNAs that are attached to amino acids. So these are charged tRNAs. You mix them all together. The ribosome bound the mRNA and the tRNAs specified by the mRNA. And in this case, this mRNA contained only one codon, GUU. So you have a solution now that has unbound tRNAs and bound tRNAs with the ribosome. You filter this. The mixture goes through the filter. The tRNAs paired with the ribosome-bound mRNA stick to the filter, whereas the unbound tRNAs pass through it. The filter was assayed to determine which amino acids were bound. And the conclusion was when an mRNA with GUU was added, the tRNAs on the filter were bound to valine. Therefore, the codon GUU specifies valine. Many other codons were determined by using this method, which leads us to the genetic code. But if you notice in the genetic code, we have all the 20 amino acids covered. But since it's a 3-letter code, there's room for some redundancy. So, for example, serine has 4 triplet codons that specifies it. Methionine only has 1, AUG. And there are also 3 stop codons, UAA, UAG, and UGA. The genetic code is sometimes called degenerate in a sense. So a degenerate code means that an amino acid may be specified by more than one codon. So you have synonymous codons, codons that specify the same amino acid. And you have iso-accepting tRNAs, different tRNAs that accept the same amino acid but have different anticodons. So now I'm going to give you an overview of how translation occurs.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Now that you've watched the video, why don't you take a break? Here is an image from Marina Abramovic's show at MoMA. The artist is present. And basically what she did was sat all day and stared at people, and they waited online to look at her. For part 2, we're going to start with an animation of translation in bacteria. And then we're going to go through it again step by step.
In translation, amino acids are assembled into proteins. Bacterial translation is initiated in 3 steps. First, the small subunit of the ribosome binds to the mRNA so that the P site is positioned over the start codon. Second, the initiator TRNA with N-formylmethionine attached binds to the mRNA through base pairing between the codon and the anticodon. Third, the large subunit of the ribosome joins the complex to complete initiation. During elongation, amino acids are joined to a growing polypeptide chain. The first step of elongation is the delivery of tRNA and its amino acid to the A site. In the second step of elongation, a peptide bond forms between the amino acids. The third step of elongation is translocation, the movement of the ribosome down the mRNA. After translocation, the tRNA formerly in the P site is now in the E site, from which it moves to the cytoplasm. Now the elongation cycle repeats itself. A tRNA and its amino acid occupy the A site. A peptide bond forms, and the ribosome translocates to the next codon. As the cycles of elongation continue, the polypeptide chain grows by the addition of amino acids to its carboxyl end. The last stage of translation, called termination, takes place when the ribosome reaches a termination codon. Release factors bind to the termination codon, causing the release of the polypeptide chain. The ribosome then translocates one last time. The tRNA and release factors exit the ribosome, and then the ribosome dissociates. Remember that several accessory proteins not shown here also participate in translation. What are the consequences of mutations in the protein coding region? Design a mutation at the highlighted base and discover its effect on the protein sequence.
So let's change this nucleotide to a G.
In this example, the point mutation causes a leucine residue to be replaced by a valine residue. Does a point mutation always lead to one amino acid change?
And the answer to that is no. Let's try the U.
In this example, the point mutation does not change the polypeptide sequence. Therefore, the mutation is called a silent mutation.
Or synonymous mutation. So let's delete a nucleotide.
In this example, the deletion of one nucleotide results in a shortened polypeptide. What are some of the other possible consequences of deleting one nucleotide from the protein coding region?
So you have a premature stop that is formed. Let's delete 2.
In this example, the deletion of 2 nucleotides results in a new reading frame that introduces one mutant amino acid and shortens the polypeptide.
Now let's just delete 3, and you'll see the consequences will be a little different.
In this example, the deletion of 3 nucleotides preserves the original reading frame, but results in the deletion of one amino acid. What are some of the other possible consequences of deleting 3 nucleotides from the protein coding region?
Well, if you deleted the stop codon, that would be a major problem because then translation would continue potentially. What about an insertion? Let's try an A.
In this example, the insertion of one nucleotide results in a new reading frame that introduces 2 mutant amino acids and shortens the polypeptide.
Let's see if we insert 2. Try 2 A's.
In this example, the insertion of 2 nucleotides results in a new reading frame that introduces one mutant amino acid and shortens the polypeptide.
Here's an image of the ribosome. The ribosome consists of 2 subunits, which are normally joined, the 30S and the 50S. If initiation factor 3 binds to the small subunit, this prevents the large subunit from binding, thus allowing the small subunit to attach to the mRNA. A tRNA charged with N-formal methionine forms a complex with initiation factor 2 and GTP. And this is what happens in bacteria. And eukaryotes, the tRNA is charged with simply a methionine, not N-formal methionine. So the complex is formed, and binds to the initiation codon, while initiation factor 1 joins the small subunit. So then you have the 30S initiation complex on the mRNA. All the initiation factors subsequently dissociate from the complex, and GTP is hydrolyzed to GDP. And this allows a large subunit to join to create a 70S initiation complex. At the end of initiation, the ribosome is assembled on the mRNA, and the first tRNA is attached to the initiation codon. The next stage is elongation. The methionine tRNA occupies the P site of the ribosome. Elongation factor 2, GTP, and charged tRNA form a complex that enters the A site of the ribosome. After the charged tRNA is placed into the A site, GTP is cleaved to GDP, and the EF2, elongation factor 2, GDP complex is released. Elongation factor TS regenerates the EF2 GTP complex, which is then ready to combine with another charged tRNA. A peptide bind forms between the amino acids in the P and A sites, and the tRNA in the P site releases its amino acid. The ribosome moves down the mRNA to the next codon. This is called translocation, which requires elongation factor G and GTP. The tRNA that was in the P site is now in the E site, from which it moves into the cytoplasm. The tRNA that occupied the A site is now in the P site. The A site is now open and ready to receive another tRNA. So at the end of each cycle of elongation, the amino acid that was in the A site is added to the polypeptide chain, and the site is then free to accept another tRNA. So how does this process eventually stop? When the ribosome translocates to a stop codon, there is no tRNA with an anticodon that can pair with the codon in the A site. Release factor 1 attaches to the A site, and release factor 3 forms a complex with GTP and binds to the ribosome. The polypeptide is released from the tRNA in the P site. GTP associated with release factor 3 is hydrolyzed to GDP. The tRNA, mRNA, and release factors are released from the ribosome. So here's a summary of how translation works in bacteria. You have tRNA charging, so the tRNAs are associated with a particular amino acid. Initiation begins where the small subunit of the ribosome binds first, and then the large unit of the ribosome binds second, through to the work of different initiation factors, and the methionine is in the P site. As elongation continues, you get the formation of a polypeptide chain. This continues until you reach a stop codon, and then the peptide is released, and you have a completed polypeptide. So in this way, amino acids are linked in the order specified by the mRNA. In eukaryotes, things are a little bit more complicated than bacteria in terms of translation. So for initiation, bacteria require a Shine-Dalgarno sequence, which is located upstream of the initiation codon. In eukaryotes, there's a Kozak sequence that is very key for initiation. Initiation also requires at least 7 factors. These factors are important in establishing the eukaryotic translation complex. The eukaryotic mRNA is a little more complicated. It has both 5' and 3' untranslated regions, as well, of course, as the coding regions with start and stop codons. It has a poly A tail. As the complex forms, proteins bind to the poly A tail, and these interact with proteins that bind to the 5' cap, which surround the ribosome, which is pictured here. This enhances the binding of the ribosome to the 5' end of the mRNA and helps facilitate translation. And now I'm going to show you a video that has translation in more detail.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Once you make a protein, they adopt a structure. So proteins are polymers consisting of amino acids linked by peptide bonds. The amino acid sequence is its primary structure. This structure subsequently folds to create secondary and tertiary structures. Then 2 or more polypeptide chains associate to form quaternary structure. So you have your primary amino acid sequence on the left, and this sequence here is forming a helix, and that is the secondary structure. This helix becomes a part of the folding of the rest of the polypeptide chain, and This causes the protein to develop its tertiary structure, which is close to the final structure of the protein. So, but in some cases, proteins that have adopted their tertiary structures can interact with other proteins to form a larger complex, and this is the quaternary structure. At this point, we have created a functional protein that can act in the cell. So translation's work is done. So here are some genetic terms and concepts I want you to remember. Sense codon, a codon that specifies an amino acid in a protein. Degenerate genetic code refers to the fact that the genetic code contains more information than is needed to specify all 20 common amino acids. Synonymous codons, different codons that specify the same amino acid. Isoaccepting tRNAs, different tRNAs with different anticodons that specify the same amino acid. Wobble, base pairing between codon and anticodon in which there is non-standard pairing. This usually occurs at the third position of the codon and allows more than one codon to pair with the same anticodon. Reading frame, particular way in which a nucleotide sequence is read in groups of 3 nucleotides, codons, in translation. Each reading frame begins with a start codon and ends with a stop codon. Initiation codon, the codon in mRNA that specifies the first amino acid, F-methionine in bacterial cells and methionine in eukaryotic cells. It's most commonly specified as AUG. Universal genetic code refers to the fact that particular codons specify the same amino acids in almost all organisms. 30S initiation complex. Initial complex formed in the initiation of translation in bacterial cells consists of the small subunit of the ribosome, mRNA, initiator tRNA charged with F-methionine, GTP, and initiation factors 1, 2, and 3. 70S initiation complex. Final complex formed in the initiation of translation in bacterial cells consists of the small and large subunits of the ribosome, mRNA, and initiator tRNA charged with methionine. Amino acyl A site, one of 3 sites in a ribosome occupied by a tRNA in translation. All charged tRNAs, with the exception of the initiator tRNA, first enter the A site in translation. Peptidal site, the P site, one of 3 sites in a ribosome occupied by a tRNA in translation. In the elongation stage of protein synthesis, tRNAs move from the amino acyl site to the P site. Here is a beautifully colored digital representation of a Drosophila embryo. Each color represents a different gene that marks the fate of each of the cells in this Drosophila embryo. In the next lectures, we will explore gene regulation in prokaryotic and eukaryotic organisms. See you next time.